all possible interaction model search is not really computationally tractable at the moment, given the sizes of the PGC2 and soon to be PGC3 data sets. But I will highlight that there are members of the upper stage that have software to perform all possible two-way interaction search in genome-wide data, and I can put you in touch with them if you send me an email after the talk. Okay, so my thought was that machine learning has been used very successfully in multiple different applications, and since machine learning was developed for use with high-dimensional data, perhaps machine learning is a good approach to detect sets of SNPs that influence case status. But the question that I have, and what I'm going to focus on during this talk, is there anything that we really need to consider before applying machine learning algorithms to the data, or can we just download an R package and just run it on our data and feel happy about the results that we get? So if you're not familiar with machine learning literature, it's full of statements such as X method does not overfit, or Y method is a good off-the-shelf classifier, meaning that you don't really have to understand the nuts and bolts of it. You can just download your package and apply it to your data and get robust results. You don't have to tune the hyperparameters that much. But often there's very little or no proof given in the actual manuscript that this is true. So what you tend to see are very small simulation studies with 4 or 5 variables, and that obviously isn't adequate evidence to really make these claims, especially given that these methods are really developed for use with high-dimensional data. So just for you who are not familiar with machine learning, there are sort of 3 flavors. There are ensemble-based methods, which are ensembles of base classifiers. So in these examples, these are trees, so random forest and boosting. There are regression-embedded methods, such as the lasso, the elastic net, Monte Carlo logic regression, and then there are kernel-based methods, such as support vector machines. Now, today I'm going to focus on random forest and similar ensemble-based methods. But I'm happy to focus on the other 2 types in upcoming talks if people have interest. Okay, so I'm going to walk you through the steps of a random forest. And so this is just so that we're all on the same page in terms of what the random forest is doing. And I'm going to talk about something called variable importance measures, which are a measure of the association between the SNPs and the outcome of interest. So in step 1, you can see I have our cases and controls here, and I'm going to select a subsample of the cases and controls, and I'm going to set the additional samples aside. So if these boxes represent SNPs, I'm going to select some subset of SNPs, and then using these 2 subsets, I'm going to start to build a recursively partitioned tree. So at this first split, I'm going to take this subset, and I'm going to test all of these SNPs to see how well they can partition the data into cases down one side of the split and controls down the other side of the split. And then once I've done that, so you can see here, the green SNP is the best SNP, SNP1. And so we can say that, do these individuals carry the big A genotype in SNP1? Yes, they go down this side. No, they get on this side. And then at this level, I'm going to take another subset of these SNPs, and I'm going to do the same thing again, over and over to build a recursively partitioned tree, which are obviously usually much larger than the tree in this example. The goal, though, at the bottom in these terminal nodes is that we want to be able to perfectly separate the cases from the controls. Sorry, in step 3, this set here, the individuals from here, this is called the in-bag sample. In step 4, what I'm going to do is I'm going to take the individuals here that I didn't use to build the tree, and I'm going to run them through each split of the tree. And so if we start at the top, I'm going to take these individuals. I'm going to see how well this split can predict whether someone is a case or a control. Then I'm going to take the genotypes at this SNP, I'm going to permute them, and then I'm going to run the individuals with the permuted data through that split again. And so the point here is to break any association between SNP1 and case status. So I'm going to take the quantity that I calculated using this split with the observed data minus the quantity that I calculated using the permuted data, and I'm going to call that a measure of variable importance for that SNP in that tree. All right, so I'm going to do that for every single split in every single tree. So again, this is a random forest, so I have to do steps 1 through 4 thousands of times to create thousands of trees. And then at the end, I'm going to calculate the variable importance by taking the average of this quantity that I calculated here in step 4. And these are in the out-of-bag sample. So these are the in-bag, these are the out-of-bag. Now, there are two sort of main flavors of variable importance measures. This one I'm going to call the permutation-based method. There is another one that is based on the in-bag samples. And so that one is what I'm going to call the Gini index-based variable importance measure. So this is the average across all the trees in the forest of the decreased in node impurity for each predictor, right? So, but this is using the same samples that we used to create the tree in the first place. And as I said, variable importance measures are the main way that we understand what's happening in the algorithm. It's the main way we can get an estimate of which SNPs are most important in terms of prediction of the outcome that we're interested in. Okay. So going back to my original question about, you know, can we just apply machine learning? Is there anything we need to worry about? The main thing here is if we're going to use these variable importance measures to determine what's going on with the algorithm, are there conditions in which the random forest variable importance measure is misleading or where the algorithm might fail? And so there are, we've identified several places where the algorithm might fail or you might get a spurious result from the random forest variable importance measure. So one is in using different scales of measurement. So in this case, you might have one variable that's continuous and you might have another variable that's binary. So they have different scales of measurement. Another situation is when you have correlation between predictors, which is obviously really important for us because we're looking at genome-wide data, so there'll be linkage to disequilibrium between SNPs. Another situation is when you have differing category frequencies. In our case, this would be where you have different minor allele frequencies. And then the final bit that I'm going to talk about is some new work that is hot off the presses, that hasn't been published, where we're looking at interaction effects and new variable importance measures that have been proposed in the past few years and see if they fare any better than the permutation-based variable importance measure or the Gini-based variable importance measure, which are the standard ones. Okay, so before I go any further, are there any questions at this time? Okay, so as I said, the first one that we're going to look at is different scales of measurement.
Wait a second, Kristin. I muted everybody. That's why nobody could ask. But I think like Eli,
I don't see any hands up, but I can't see any hands.
I see at least Eli on the video. He's trying to.
All right.
I'm trying to fix my.
Eli is here. Okay, right. Eli.
No, I don't have any questions. I'm just trying to fix my computer.
Oh, okay.
I'm using my desk.
Somebody else here who wants to ask something because I muted everybody. I'm sorry for that. So, do we have anybody? No. Okay, then it's fine. Sorry, Kristin. I didn't want to interrupt. But.
Okay, so this first study that I'm going to talk about by Carolyn Strovel et al. Examined random forests, which I've just talked about, plus an additional random forest type algorithm called conditional inference forest. So the difference between these two types of random forest is that the random forest splitting criteria is based on the Gini index impurity, which I mentioned before, whereas the conditional inference forest is based on a chi-square or a t-test, and you can actually adjust for multiple testing because every time you do a split, you're going to test all the SNPs that you've selected for that split, and you're going to test all of the different cut points that you might have. So for a genotype, you'd have 2 cut points, right? So, so what she showed here, she did a very simple simulation, and again, this is fairly typical for machine learning type papers where the simulations tend to be trivial. They tend to be very small. But what she's showing here is a simulation study where she looked at different scales of measurement, and then she also looked at the use of bootstrapping when you select that original set in to grow the tree versus subsampling. And so what I'm showing here is what she found under the null. So in this case, none of the variables are associated with the outcome. All right, so we shouldn't see anything different between different variables because none of them have an association with the outcome. So she simulated 5 variables, and you can look, she's listed the distribution here, but essentially on the x-axis, she's plotted x1, which is standard normal, x2, which has 2 categories, x3 that has 4 categories, x4 that has 10 categories, and x5 that has 20 categories. And so on the top row, these are for random forest, and on the bottom, this is for conditional inference forest. And so these are selection frequencies. So this is how often the variable gets selected into a tree. And you can see that the variables such as x1, x4, and x5 that have more cut points are selected more frequently. And this is due to, obviously, multiple testing. And so that's true also for conditional inference forest when you use bootstrapping. However, when you use subsampling, this still is a problem for random forest, but it is not a problem for conditional inference forest. And I'll get back to that in just 1 minute. So I mentioned before that the Gini index-based variable importance measure is based on this sort of Gini index splitting criterion, right? And you can see here that this is, again, under the null. And so what we've got here plotted on the y, so it's, again, x1 through x5. On the x, on the y, we've got the Gini index-based variable importance measure. And again, this is under the null. So we shouldn't see any difference whatsoever among these 5 variables because none of them are associated with the outcome. But again, you can see that these plots look very similar to the selection frequency plots, right? Because every time a variable is selected into the model, it adds more to the Gini index-based variable importance measure. So again, this is random forest, and you can see that no matter if you're using replacement or not, you still get a situation where you get spurious results. So here, this is the variable with 20 categories, and you can see that the Gini index-based variable importance measure is much higher than the one that's here, x2, which is binary. So clearly this is a problem. It also leads to low power under the alternative hypothesis, where she assumed the variables to be associated with the outcome. Okay, so when they looked into this a little bit further, what they found is that, so these are just chi-square tests, and they were looking at what happens before bootstrapping and then after bootstrapping. And so these are just for the variables that are categorical variables. So again, this is binary, 4 categories, 10 categories, 20 categories. So the chi-square tests under the null should be distributed uniform 0, 1, right? And so before bootstrapping, you can see that the median is set at 0.5, and the interquartile range goes from 0.25 to 0.75 for all of these variables. And again, it's not the best way to plot this, to be honest, but essentially this is a uniform distribution on 0, 1, which is what you expect under the null. But after doing the bootstrapping, you can see that the p-values, which are plotted here, you can see that for x5, which is the variable with 20 categories, the p-values are much reduced. And again, this is under the null, so we shouldn't see this. This is a problem. Whereas the binary one is slightly reduced, but not as much. And so in their simulation, they had 120 observations that they simulated. So with larger sample sizes, it won't be as much of a problem. But the issue here is that when you do the bootstrapping, you have some individuals who will not be part of the bootstrap sample, and some individuals who will be in there more than once. And then on top of that, there's also an issue with multiple testing because you're testing 19 cut points here, but only 1 here. Okay, so just to recap that section, the use of predictors with different scales of measurement leads to increased selection frequencies in random forests due to bootstrapping and multiple testing. Conditional inference forests with subsampling can improve the performance of the Gini index variable importance measure, but they suggested that we not use the Gini index-based variable importance measure and instead stick to the permutation-based variable importance measure, which isn't affected by these problems. Okay, right, so are there any questions at the end of this bit? I can't see if anybody...
Yeah, so if you have to unmute yourself just to be sure here.
Okay, so if there are no questions, I'm going to go ahead. So the other issue that we have in genomic data that may be a problem is we have linkage disequilibrium between SNPs. And so my question again is this going to be an issue for the use of this particular algorithm with genomic data? So I'm going to talk about one of the papers that I published on this back in 2009. And so what I'm doing is I'm looking at within predictor correlation and whether that has an effect on variable importance measures. So I conducted a simulation study that assessed the impact of correlation on variable importance measures under both the null and the alternative. So part of the simulation study was a genetic study where I simulated 5 genes based on, if I remember correctly, it was HapMap phase 1 SNPs that I had phased and then simulated a model onto the haplotypes from the genes, 199 SNPs in total, 500 replicates per condition. And of course, because I'm using the haplotypes, I retain the original LD structure. So these plots are a little messy, and I apologize for that. So in plot A, what I'm showing is our box plots. So each one of those little things is a box plot for an individual SNP going across the different chromosomes, because they were on different chromosomes, but you can see there are 5 genes. And so this is the Gini index-based variable importance measure. This is the permutation-based one. The line here represents 0, and the line here represents 0. And so this is under the null. So again, there should be no association between any of the variable importance measures and the outcome because it was simulated to be under the null. And again, these are box plots of the variable importance measure for each SNP across 500 replicates of the simulation. So what you can see in plot A, which is the Gini index-based variable importance measure, is that there are box plots that have really large variable importance measures up here, and then there are sets of SNPs that have very low variable importance measures, excuse me, down here. So if this were real data and you didn't know the answer, you might think, wow, I have a really nice hit over here, I'm going to follow that up. And you might not want to look at these because they have the lowest variable importance measures. This is the permutation-based one. And again, this line is at 0, which is the quote-unquote right answer. And you can see that all of these box plots are centered on 0. But what I can tell you about these box plots up here is that these are for SNPs that are not in LD with any other SNPs. These are SNPs that are simply uncorrelated with anything else. And so this is an LD plot of one of the genes. That's the biggest gene. That's the one in the center here and in the center here. And what I've shown, I've got a box around this block of LD here. There are about 30 SNPs in that block of LD. And you can see it's pretty tight LD. This is our square. And so this box corresponds to this section in this plot and this section in this plot. So you can see that when variables are correlated, it artificially reduces the variable importance measures in the Gini index-based variable importance measure, whereas the ones that are not correlated are inflated. For the permutation-based one, even though there's less variability, you're still getting the right answer. You're still finding that there's no association, which is the true answer here because it's under the null. So then what happens under the alternative? So this is a plot of the Gini index-based variable importance measure. This is a zoom in of the plot that you saw before. Again, these are box plots of the Gini index-based variable importance measure for each SNP across 500 replicates. And this is a zoom in of the area around this LD block. And so this line represents the SNP that was simulated to be associated with the outcome with an odds ratio of 2. So an odds ratio of 2 in what we do is a huge odds ratio, right? And so you would assume that this method should hopefully be able to find this signal, but you can see that there are box plots that have, I can't read the y-axis, but that have much higher variable importance measures, right? And so those are ones over here that are not in LD with anything else. And the true signal that was simulated here on this SNP, which is part of this block, has a very low variable importance measure. So we would never follow that up, even though that is the true signal. And that is, again, because of the correlation between the SNPs in that area. So if you think about why this would occur, so if you remember from the original diagram that I was showing you about how a random forest works, you know, every single time that you are going to create a split in the tree, you're going to take something called M try, so the number of variables you're going to try. So you're going to take some random set of SNPs at the top, and you're going to test them. And you're going to pick the one that is the best, that performs the best split, right? So let's say SNP 1 from this block here was selected at the top of the tree. So then you go down the tree, and then at the next level, or the level after that, you take another random subset of SNPs, and let's say SNP 2 in this block as well is selected into there. So if you split on SNP 1 at the top and then split on SNP 2 further down the tree, obviously most of the information is contained in SNP 1. So the actual variable importance measure for SNP 2 for that tree is going to be reduced because most of the information is contained above it. But because we're randomly selecting subsets each time, there will be some situations where SNP 1 is at the top of the tree and SNP 2 is below it, but other situations where SNP 2 is at the top of the tree and SNP 1 is below it. So then when you average across the tree, it actually reduces the value for the variable importance measure for the ones that are correlated. Okay, so just to recap that, within predictor correlation leads to spurious results for the Gini-based variable importance measure, both under the null and the alternative. The same correlation leads to lower variability in the calculated variable importance for the permutation-based measure, but it doesn't lead to spurious results. I think that's the end, yeah. So are there any questions about that section? Okay, so moving on. So the next section is about different category frequencies. So in the simulations that I'm going to present, I just had a binary variable. So I have a major and a minor category frequency, but this is also true for minor allele frequencies for SNPs. And there was a paper, a follow-up paper that was published that looked at genetic data and found what I am about to show you here with the binary variables. Okay, so going back to the first study by Carolyn Strobel et al., she had shown that variables with different scales of measurement result in spurious results for the Gini index-based variable importance measure. This paper here that was published in Briefings in Bioinformatics said that, well, if we have SNP data, we don't have different numbers of categories, right? Because we have 3 categories for the most part for every SNP. So we don't need to worry about that anymore. We can use the Gini index-based variable importance measure and not have to worry about spurious results. And then I wrote a letter in response to that, and so I'm going to talk about those 2 papers now. Okay, so in the original paper, what we have here are 2 plots. So they had real data on about 800 SNPs. And so what's plotted here is the Gini-based variable importance measure. This is the permutation-based variable importance measure. So what they'd done is they had run random forest once on the full data set, and they plotted the rank of each SNP, and that's what's plotted on the X for both of these, okay? Then they had taken 100 subsamples, 90% subsamples of the data, and they had rerun random forest on the 90% subsamples, so a sort of jackknife type procedure. And then for each of those subsamples, they're plotting on the Y the rank in the data set with 10% of the observations not included. And so what they concluded from these 2 plots is that you can see there's much more of a linear relationship here, sorry, using the Gini index-based variable importance measure than using the permutation-based one. And so they concluded that the Gini index variable importance measure was more stable and should be preferred when you're looking at genotype data versus the permutation-based one. So, sorry. What I found, curious about this plot is that, so if you look at the rank starting at 0, okay, so obviously 1, so this is the top ranked SNP, and second, and so forth. you can see there's a pretty tight correlation at the top of the list. So you would assume that these would be SNPs that are fairly strongly associated with the outcome. And that's true in both the Gini index-based measure and the permutation-based measure. You can see there's a pretty strong correlation at the top of the ranked list for this one as well. But what I couldn't quite understand is why there's a pretty tight correlation at the bottom. Because, I mean, I can't remember what it was some sort of cancer, I think. I can't remember what their outcome was, but it was a complex disease. It wasn't something where there are loads of really strongly associated SNPs. And so I couldn't figure out why this would occur here, whereas here it kind of looks like random scatter. So I performed a synthetic simulation with 1,000 cases and controls. Now, there's no correlation between any of the variables in the simulation. And so to try to replicate what they had shown, I set the first 5 variables to be associated with the outcome. So they had odd ratios of 3, 2.5, blah, 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 down to 1.25 with the minor category frequency of 0.5. Remember, these are all binary. So then I had 45 additional variables that I simulated that have no association with the outcome. So the first 5 were under the alternative. The last 45 are under the null, so a total of 50 variables. So then I have 2 different conditions. And so in condition 1, I made the minor category frequency for the 45 variables that are not associated all equal at 0.5. And then in condition 2, I varied the minor category frequency from 0.5 down to 0.1. So there are 5 variables in each of these. So 5 with a minor category frequency of 0.5, 5, 0.4, so forth and so on, down to 0.01. And then I performed the same analysis as in the original paper where I ran random forest once on the full data set, and then I took 100, 90% subsamples of the data and plotted those, as I said, just like in the original paper. So what I can show you here is at the top, these are the Gini index-based variable importance measures. These are the permutation-based variable importance measures. In this column, the minor category frequency for the 45 unassociated variables is set equal at 0.5. And then here what I've done is I've varied, this is condition 2, I've varied the minor category frequencies from 0.5 down to 0.001. The first 5 variables, as I said, are simulated under the alternative with odds ratios ranging from 3 to 1.25, I think. And you can see that in each case, those are picked up, right? Those are picked up. And just to remind you, this is the rank in the original data set. These are the ranks in the 190% subsamples for all the plots. But you can see that when the minor category frequency is equal at 0.5, there is somewhat of a linear relationship, but there's much more random scatter here versus here, where the category frequencies vary. And I can tell you that this block of 5 variables are the variables with the lowest minor category frequency, 0.001. These 5 are the ones with minor category frequency 0.01, these 0.05, and so forth and so on. The permutation-based variable importance measure, again, you get the random scatter here, which is similar to what you see with the Gini, but you don't get as much of a strong linear relationship with these variables that have low minor category frequencies, or in our case, they would be low minor allele frequencies. So again, this is the original plot for the Gini-based variable importance measure, and then these 2 are the plots from my simulation. And you can see that this plot looks a lot more like this plot, and so these are very likely to be simply SNPs that have very low minor allele frequencies, essentially. So I can also say, so again, the original paper had suggested that the Gini index-based variable importance measure was much more stable than the permutation-based one. But in the paper that I reference, where I responded to that paper, I show situations where actually the permutation-based variable importance measure is more stable when you have strong correlation between SNPs or between variables than the Gini index-based variable importance measure. So essentially, even though permutation-based variable importance measure can be unstable, they are robust to correlated predictors. So what I do in practice is I simply rerun random forest 500 or 1,000 times on the data set using a different random number seed. And then I take an average of the variable importance measure for each variable over all of those rerunning, all the times that I've rerun, in order to get a stable estimate of the variable importance measure. I think I've shown several times now that Gini-based variable importance measures can lead to spurious results. And so the next section, I'm going to talk about new variable importance measures and how they might fare under the null and under the alternative that includes interactions. So at this point, are there any questions?
So 1 second. So can we go back to the correlated SNPs in there? So this is the section before, actually. So what I don't completely understand is why don't these methods actually like try to account for correlations like those? So why don't they, for example, remove any dependency beforehand, for example, so like clumping the results beforehand or something like this to get rid of this phenomenon?
So there are a couple of approaches that have been proposed. Obviously, in practice, you would want to clump your data or LD prune your data before using one of these methods. So there have been a couple of methods that have been proposed. One I'm going to talk about in the next section, which, I'll talk about in the next section. The second one is a paper by Yan Meng that came out 2009 or something, and I don't know if there's software available for it, where she essentially took the LD matrix between all of the SNPs, and then given each set that was selected in, and whatever variable was selected to split on, then there was sort of a lookup to see, okay, we're going to throw away any SNP that's in LD at some level with the SNP that's in the tree, and then take a random sample from what's left.
Mm-hmm.
Which I think is a great idea, but I don't think that there's software that's available for it. So there's a software called Random Jungle that was developed by Andreas Ziegler and colleagues at the University of Lübeck. And so it says that it does the method that was developed by Yan Meng, but actually it doesn't. It doesn't actually do all of the stuff that is in the paper. So it doesn't actually select subsamples based on the LD matrix. So it's called the Meng variable importance measure, but actually all it's doing is it's dividing, when you take the average across all the trees in the forest, it's not dividing by the total number of trees, it's only dividing by the number of trees that SNP was in. That's the only thing it actually does. So It's a little bit confusing, but it doesn't actually do what it's supposed to do.
Okay, so you're saying like, so there are definitely approaches who are trying to get this phenomenon under control, but it's not like done everywhere.
No, and I mean, essentially, as I said, the easiest thing to do is just to LD prune. There's another way of doing this that one of my students has been working on. It was published actually a few years ago by Lübke, who's at the University of Notre Dame. And so what she had done is create subsets of SNPs that are in linkage equilibrium. But then the problem there is, how do you combine across these different subsets of SNPs that are in LE? And that becomes more difficult as the methodology that you're using becomes more complex. So you would have, 10 or 20 or, however many subsets of SNPs from your GWAS that are in LE. So essentially what she was trying to do is prevent throwing out information by LD clumping or by LD pruning or by clumping.
Yes.
But then when you get to the end, you have 20 or even 50 data sets of SNPs in LE that overlap with each other. How do you combine your estimates at the end of that is the question. So let's say even something like a polygenic score. Right, so you run your polygenic score on all these LD, independent subsets, but then what do? Do you take the average? I mean, how do you combine them across these different sets?
What do you mean exactly with different sets? So
what she had created was a way of So she's taking they're not random, sorry. She's taking subsets of SNPs.
Yes.
And so for each subset, it's a set of SNPs that are in LE.
Yes.
Okay? But there's going to be overlap between the different sets, but every SNP will be included in at least one set. So she doesn't want to throw away any information. She wants to have every set, every SNP in at least one set so that you can have all the information. But then if you have 20 or 50 subsets of SNPs that are in LE, how do you combine your estimates at the end?
Well, it's probably difficult. So definitely. So usually, what we do in our polygenic risk course, because you asked about that, we do not want to we do not try to keep all this information, all the information.
Right. No, I know.
To that. So exactly. Yeah. Okay.
Yeah. So it is a problem, and it's something that needs to be fully addressed, but so far hasn't really been adequately addressed outside of doing something like clumping or LD pruning.
Yeah. Okay.
So, sorry, this is Raymond. I can probably comment as an author on the Lubke paper.
Oh, right.
So the proposed solution to that issue, at least in the paper, is that you take the mean importance of the SNP across subsets that it's included in.
Yeah, so I actually haven't read that paper in a while, sorry, because I didn't think it would come up today. But I mean, it's a valid way of doing it. But then again, if you're looking at more than just a variable importance measure, maybe you want to do a different type of analysis, or as I said, like a polygenic score, then you just take a polygenic score, just take an average. I mean, it's not 100% clear the best way to handle the LD situation.
Yeah, I fully agree on that. If your goal is prediction rather than the variable importances, then that's an open question.
Yeah. Okay. All right, so I'm going to move on to the last bit. So, so in this last bit, this is, as I said, very new work, so not published and preliminary. So it's work that's been led by my PhD student, Marinera Gonzalez, who you can see here. So here we wanted to look at an expanded set of variable importance measures. So in random forest, we use the ones that I've been talking about throughout this talk, the standard Gini and permutation VIMs, plus scaled permutation VIMs, two different types of scaled VIMs. And then in random forest, we also looked at a conditional permutation VIM. In conditional inference forest, we use the standard permutation one, and then one based on the ROC area under the curve, and then also minimum depth. So I'm going to talk about the new ones now. I'm not going to go over it there. Okay, so the scaled VIMs are just a permutation-based variable importance measure that's been scaled by an empirical estimate of the standard error, and there are two different definitions of this. But these tend to not be used as much because obviously if you have a large number of trees, the standard error is going to be smaller than if you have a small number of trees. So the number of trees confounds this scaling. So they don't tend to be used, but we included them for just thoroughness. Okay, so the conditional inference forest permutation one is the exact same as the one I discussed for random forest, but using a different type of random forest, which is the conditional inference forest. Also in conditional inference forest, you can get an ROC area under the curve VIM, which is the exact same idea as the permutation-based VIM, but what you're doing is you're calculating the AUC before and after permutation. You're taking the difference in the AUC value instead of looking at the prediction, in the out-of-bag samples. All right, so going back to Stefan's question, there was a paper that came out a few years ago now where they proposed a conditional variable importance measure. So this conditional variable importance measure essentially creates strata of variables that are correlated with the variable of interest. And then it permutes the variable of interest within the strata. And so this was designed to adjust for correlation between variables in your data. And then the minimal depth variable importance measure. So this is, again, one of the trees that I showed you earlier. All it is the average across the forest of the minimal depth of the predictor in a tree. So if the green one was the one that we wanted, this would be given a minimal depth of 0 in this tree. If it was this blue one, it would be given a minimal depth of 1. So it's the first occurrence in the tree. So if there's a green one down here at level 6, it's still going to get a value of 0 because it's at the top. And so the idea here is that variables that are more strongly associated with the outcome will tend to be selected into the top of the tree more frequently. They're more influential than variables that are not associated or not as strongly associated. Okay, so in this, we have, we looked at simulations under the null and under the alternative, 500 replicates. We have 100 variables, and what I'm showing today are results from continuous outcome and predictors. We've also done this with binary outcomes and predictors, and there is no difference. So what I say holds true for all of the simulations that we've done, but there was too much to go through all of it during this talk. So we varied the number of correlated predictors. So in one set it was 5, in one set it was 20, in one set it was 40 out of 100 variables. And then also the strength of correlation between the predictors. So some with correlation of 0.1, 0.4, and 0.8. So in total, 9 different conditions. And yeah, so we allowed the number of variables that we selected each split to vary for the minimal depth, because previous studies had shown when there's correlation, you should use a smaller value. Okay, so this is our generating model here, and it's just a very simple regression model where variable 2 and variable 90 have a main effect and also an interaction effect with an error that's pretty large, so this is relatively weak association. So again, I just want to point out variable 2 and variable 90 are the ones that under the alternative we're interested in. Okay, but I'm going to start out with what happens under the null. And so in this case, I know these plots are difficult to look at because they're pretty tiny. So in this case, this is under the null. We have 5 variables correlated with a correlation value of 0.1. So this is the weakest correlation condition. In each plot, we have a different variable importance measure. On the y-axis is the value of the variable importance measure. On the x-axis are the numbers of the different variables. So in each of these plots, what you're going to see is the first 3 variables are part of the correlated set. And the last 2 variables are part of the uncorrelated set. This is just for ease of viewing. If you plot all of the variables that are correlated and all the variables that are uncorrelated, there's no difference. So we just selected these to show, instead of the plots from earlier where I had plotted lots of things and it was difficult to see. All right, so as I said, the first 3 are in the correlated set, the second 2 in the uncorrelated set. This is under the null, so we shouldn't see any difference across these different sets. And so this is the Gini-based variable importance. This is the permutation one. These 2 are the scaled permutation. This is the one that is for correlated variables, that is based on permuting within strata of correlated variables. This is the variable index measure for permutation from conditional inference forest, and again, conditional inference forest using the area under the curve. And then these 2 are the minimal depth. So what you can see from these plots is that there's no difference between the different variables, no matter if they're correlated or uncorrelated. They all give you pretty much the right answer, that there's no association. But interestingly, this set here, there's no variation. And so we wondered why that could be. And so we're going to see this over and over again, that there's not much variation. So I'm going to come back to that later. All right, so here is in the strongest correlation condition where we have 40 variables correlated with a correlation of 0.8. And again, this is under the null. You can see that, as I reported earlier, the Gini index-based variable importance measure reduces the importance of the variables that are correlated, inflates the ones that are not correlated. Now, you can't really read the y-axis probably very clearly given the size of this, but these values are pretty small. There's not much difference, but you can see from the box plots that there is a significant difference where the permutation-based variable importance measure actually gives more importance to the correlated variables and less to the uncorrelated ones. Same thing is true when you scale them. I'll come back to this one. The ones that perform the best are the ones from conditional inference forest. So this is the permutation-based one, the AUC-based one, and they pretty much give you the right answer. There's not significant differences between the variable importance measures under the null, no matter if they're correlated with each other or they're not. Again, significant differences between uncorrelated variables and correlated variables for the minimal depth, with the uncorrelated variables here showing that they're higher up the tree than the ones that are correlated with each other. And again, going back to this one that was designed to be used with correlated predictors, it does give you the right answer. This is center non-zero. This is under the null. But you can see there's no variation whatsoever among the ones that are correlated, and there's normal-looking levels of variation among the uncorrelated ones. So the uncorrelated ones are obviously just the standard permutation-based measure, right? Because we're not conditioning on anything, because they're not correlated with anything. Okay, so this is under the alternative, and that was the generating model that I showed you earlier. So V2 and V90, variable 2 and variable 90, are the ones that are associated and interact. So those are the first one and the last one in each pot. And you can see here that when you only have 5 variables that are correlated at weak levels of correlation, all of the methods are able to find this signal. It's not a difficult signal to find, except the one that's based on conditioning. And so what we found, so when you set up this method, this variable importance measure to run, you have to give it a correlation cutoff value to create the strata. And so because our correlation is low at 0.1, we set the cutoff to be 0.05. And what ends up happening is that you get some spurious correlation between the ones that are not supposed to be correlated that can be at 0.05. And so it's actually conditioning on a whole set of variables, the 5 variables that are correlated at 0.1 plus additional ones. And so the number of strata becomes very large. So when you permute within the strata, you're not actually doing much of anything because you may only have 1 or 2 observations within that individual strata. And so that's why there's no variation whatsoever. And so finally, we get to the situation where we have the highest level of correlation, and we have again, the generating model with variable 2 and variable 90 being associated with the outcome and interacting with each other. And we have the exact same set of variable importance measures. So you can see V2 in almost all of these conditions. It's difficult to find it for all of the variable importance measures because of the tight correlation and the large number of variables that are correlated with it. But you can see that some methods actually perform a little bit better than others. So here's the permutation-based variable 2. These two are correlated with variable 2 at a correlation value of 0.8, so they're tightly correlated. I wouldn't necessarily call these false positives because clearly they're tagging the true variable that's associated, which is V2, so that's all right. And they're significantly different from the one that's not associated. Whether or not we could find the actual V2 signal among the correlated variables, I'm not sure, but at least we're picking up the right answer. The Gini index-based variable importance measure for variable 2 is lower, significantly lower than this variable here, which is not correlated with anything else and also not associated. So again, we'd get the wrong answer. The 2 variable importance measures based on conditional inference forest actually perform slightly worse. So these are all not significantly different from one another, even though this is the truly associated variable. This one's not associated and not correlated, so we wouldn't find it. Minimal depth, again, the correlated variable importance measure is reduced versus the one that's not associated and not correlated. And again, the one that was designed to be used with correlated predictors, we wouldn't be able to find any signal because the number of strata becomes so large because we have 40 variables that are correlated. And so when you do the permutation within the strata, you're basically not really permuting much of anything. So there's no variation there. Okay, so just to recap, under the null with a small number of variables correlated and low correlation, all of the VIMs except the conditional one with no variation performed adequately under the null with a large number of correlated variables and high correlation. The Gini index, the scaled permutation ones, conditional minimal depth produce spurious results. But under the alternative, all of the variable importance measures were able to detect the true signal in the small number of correlated variables, low correlation condition, except that conditional one. But when you have a large number of correlated variables and high correlation, all methods could detect the uncorrelated interacting predictor, but they varied in their ability to find V2, which is the correlated interacting predictor. All right, so just to close, the work on random forests is fairly extensive, but unfortunately, most of the machine learning algorithms haven't had this type of attention. And so most of them require further study to understand better when they might fail or when they might give you spurious results. So one of the goals in the epistasis working group that I'm chairing is to perform similar simulation studies to compare different algorithms and different methods across these types of conditions, which I think would be really useful across not just the PGC, but across genetics in general, because as I said, most of this work has not been done. Epistasis working group is open to anyone who's interested, so email me if you'd like to join the Google group. And just to close, I want to thank my student, Laura Nera Gonzalez, my funding, and my email is there if anyone would like to email me about the slides I've presented or about the epistasis working group. So I'll take any questions. Sorry, Stefan, you're on mute. There we go.
I just realized that. So thanks so much, Kristin.
I could see you talking. I'm like, oh no, did I just finish all of that and nobody could hear me?
No, that was just me. So okay. Thanks so much, Kristin. It was really wonderful to see all that. So we're very close to the hour, so yes. Almost no time left. Anybody here has some pressing questions? Otherwise, I mean, Kristin's email is there. I mean, I think we all would be very interested how this all behaves like under real data
yes.
Scenarios, right? So like when we go into PGC data or similar stuff, or maybe some easier stuff like Crohn's disease or so, where we can see like this in the real world and so on. We probably don't have time to discuss this at length here. Maybe in a second session, or I'm not so exactly sure where we want to go from here, but I think that's probably to discuss internally.
So essentially, as I mentioned, ideally, what we plan for the epistasis working group is for there to be simulated genetic data for people to apply their methods to. And then, you know, it's sort of like one of those competitions where someone does a simulation and they keep the results, you know, not the results, but the true model, they keep it hidden. Everybody performs their method, and then at the end they all contribute, and then you see which one performed the best. You know, it's that sort of thing that I think is really necessary in using these methods, because we really don't know. We don't know which ones are going to perform well. We don't know which ones are not going to perform well or be confounded by things that are, you know, standard in genetic data, like linkage to disequilibrium. So.
Okay, so and this epistasis working group is led by you, and and so there is a Google group for that, apparently,
yes,
right?
There is. Yes, there is.
So we're even over the hour right now. So 22 people kept quiet for an hour, but I think they all listened. So for everybody, please also add your name to this Google sheet that Krista sent around. And again, let me thank Kristin, who did a wonderful job to keep to the hour and to tell us some interesting details about methods that we do not apply on a daily basis. That's for sure. So at least I don't, and it's very, very, very commonly asked about machine learning here. So I think it's very good to have you on here. And
right, great. Thank you for having me.
Thank you all, and I will close this now, and I wish you a good afternoon or morning or whatever you have. Thank you.
Good afternoon. Bye-bye.
Bye-bye.